this slide shows a little more detail about folliculogenesis. I told you earlier that folliculogenesis is basically the process of development of Graafian follicle and ultimately the release of the oocyte and formation of corpus luteum. So each oocyte is enveloped by primordial follicles consisting of single layer of granulosa cells and less organized layer of mesenchymal thecal cells, and these granulosa cells, they keep on dividing repeatedly and increasing the number of layers, and these concentric layers are formed around the oocyte. This proliferation of granulosa cells is mediated by paracrine factors GDF9 and a member of TGF family. Throughout this growth period, the oocyte remains in a decade stage. That means it remains in the prophase of meiosis 1, and fully grown follicle thus contains a large oocyte which is surrounded by several layers of granulosa cells, and the innermost layers of these cells will stay with the ovulated egg forming cumulus. Now when we will doing fertilization, I will tell you that this cumulus plays a very important role, because the granulosa cells surrounding the egg, which basically are termed as cumulus, they uh, send signals to the sperm which enables fertilization. In addition, during growth of follicle, the antrum cavity forms and becomes filled with a complex mixture of proteins, hormones and other molecules. So just as the maturing oocyte synthesize paracrine factors, that means oocyte as well as follicular cells, both of them synthesize certain factors and each of them coordinate each other's activity. Follicular cells will control the growth of oocyte, and oocyte will in turn control the growth of follicular cells through secreting certain paracrine and juxtacrine signaling proteins. The oocytes are maintained in a dictate stage by ovarian follicular cells, and the release from this stage and reinitiation of meiosis is driven by luteinizing hormone from the pituitary. The pituitary hormone is received by granulosa cells of the ovary, and granulosa cells in turn send paracrine and juxtacrine signals that induce activation of mitosis promoting factor in the oocyte. Meanwhile, the growing oocyte is actively transcribing genes whose products are necessary for cell metabolism, for oocyte-specific processes, and for early development before the zygote-derived nuclei begin to function. That means the oocyte has to store all the proteins, all the enzymes which are required for the initial development of the zygote, because zygote's nucleus begin to function a little later. Before that, all the necessary material for growth is provided by the oocyte. In mice, for instance, the growing diploidine oocyte is actively transcribing the genes for zona pellucida proteins ZP1, ZP2 and ZP3. These three proteins are very much essential and they should be present on the zona pellucida egg. Only then the sperm will be able to fertilize the egg. If the egg don't have these three proteins ZP1, ZP2 and ZP3, then that egg won't be able to be fertilized by the sperm. So next we have a slide showing differences between oogenesis and spermatogenesis. Now, oogenic meiosis in mammals differ from spermatogenic meiosis in numerous ways. First, when the primary oocyte divides, its nuclear envelopes break down and metaphase spindle migrates to the cortex. Now, this is very important. In oocyte, the spindle migrates to the periphery rather than remaining at the center. Why? Because you know that in both meiosis 1 and meiosis 2, the cells which are formed are of unequal size. You have first polar body and second polar body which have a limited amount of cytoplasm, and all the cytoplasm goes into the egg. So that difference in size of the resulting cells is due to shifting of spindle from center to the periphery. Fine. At telophase, both daughter cells now contain a haploid nucleus. One contains very little cytoplasm while the other retains nearly the entire volume of cellular constituents. The smaller cell becomes the first polar body and the larger cell is referred to as secondary oocyte. This asymmetrical cytokinesis is directed through cytoskeletal networks composed chiefly of actin that cradles the mitotic spindle and bring it to the oocyte cortex by myosin-mediated contraction. The similar unequal cytokinesis takes place during second division of meiosis, and in this second division of meiosis also the spindle shifts towards the periphery. Now the second important difference between oogenic meiosis and spermatogenic meiosis is that in spermatogenic meiosis the spindle is formed by centrioles, whereas in case of oogenic meiosis the spindle is formed by microtubules. Why? Because oogenic meiotic spindle lacks centrioles, because oocytes don't have centrioles. Rather, microtubules of the meiotic spindle are organized by mRNA and enzymes located on the chromosome and spindle fiber themselves. Instead of two centrioles, numerous microtubule organizing centers form around the nuclear envelope at first meiotic prophase, and these spindle then join together once the nuclear envelope breaks down. So apart from oogenesis in mammals, we have to do oogenesis in amphibians and insects also. So if we talk about frog, then in frog oogenesis takes 3 years. That means it takes 3 years for an egg to develop completely. First 2 years uh, the growth is gradual, slow, whereas in the third year there is rapid accumulation of yolk in the oocyte, causing egg to swell to its characteristic large size. Egg matures in yearly batches, with the first cohort maturing shortly after metamorphosis. The next group matures in year later. And as I have mentioned already, in the last year, in the third year, the egg increases in size tremendously, and this increase in size is because of vitellogenesis. Vitellogenesis is accumulation of yolk proteins uh, when the oocyte reaches the diplotin stage of meiotic process. And why this is necessary? Because as you know that sperm hardly carries any cytoplasm, so all the RNA, all the proteins, all the enzymes which are required for the initial development of the zygote after fertilization has to be supplied by egg itself. So this vitellogenesis, this process allows the accumulation of all the materials which will be required by the egg after fertilization for further development. The major yolk component of the frog egg is 470 kilodalton protein called vitellogenin. It is not made of the frog oocyte but is synthesized in liver and carried by bloodstream to the ovary. So in case of amphibians also, like we saw in mammals, there is an arrest in the meiotic division. You saw that primary oocyte got arrested in prophase of meiosis 1, and then secondary oocyte got arrested in uh, metaphase of meiosis 2. Same is the case in amphibian. There are certain stages which are arrested, and the division completes only if certain signals are given to the egg. Like the amphibian primary oocyte can remain in diplotin stage of meiotic prophase for years. Resumption of meiosis in amphibian oocyte is thought to require progesterone. This hormone is secreted by follicular cells in response to gonadotropic hormones secreted by pituitary gland. And within 6 hours of progesterone stimulation, germinal vesicle breakdown occurs, microvilli retract, and nuclei disintegrate, and chromosome contract and migrate to the animal pole and begin division. Fine. So in amphibians also, at diplotin stage of meiotic prophase, the development of the egg is arrested, and that development will proceed only if the stimulus is provided by progesterone. Second arrest is also there, and that arrest will be removed only if fertilization takes place. Fine. Now this diagram shows two blocks that occurs in case of amphibians. We have this first block and then we have a second block. See, we have a mitotic arrest, first meiotic prophase. This is arrested and this is termed as diplotin block. And why is this block? Because there is no active MPF. MPF is mitosis promoting factor. Fine. And this block is removed only when progesterone is present. This progesterone helps in secretion of CMOS, and this CMOS basically helps in converting inactive mitosis promoting factor to its active form by phosphorylation. Fine. The phosphorus group add होकर inactive MPF becomes active MPF, and MPF in turn is form of two subunits. We have P34 and cycline, and these subunits join with each other and with the phosphate group only if CMOS is present, and CMOS will be present or CMOS will be secreted only if progesterone is present. So this block is removed and first meiotic metaphase and then first meiotic division is completed. Now the egg enters into second meiotic metaphase, and this is again arrested at this. Now you can see this block here. Fine. And this block will be crossed only if CSF, cytostatic factor, is degraded, because CSF is basically blocking the egg from crossing this barrier, and so the egg remains in second meiotic metaphase. And how this CSF is degraded? It is because of fertilization. Once the egg is fertilized, influx of calcium takes place, and this calcium then binds with colmogylin protein, and this colmogylin protein in turn activates two proteins, CAMPK2 and calpin2, and both these proteins in turn help in degrading CSF, and which will ultimately release this block, and meiosis 2 will be completed. So one more thing which you should learn or which you should remember about oogenesis in amphibians is the presence of Lamb-Rush chromosomes. Lamb-Rush chromosomes are also termed as giant chromosomes. And why they are termed as giant chromosomes? As I've already mentioned, before fertilization, the egg has to accumulate all the proteins, all the enzymes which will be required uh, by the zygote immediately after fertilization. And in order to synthesize all those proteins and enzymes, few of the chromosomes of the developing oocyte, when the oocyte was in diplotin stage, few of these chromosomes, they unfold in a unnatural manner. They unfold in an unnatural manner, and they resemble a lamb brush. Lamb brush is basically a brush which usually is used to clean bottles. You must have seen that, and that's why these chromosomes are termed as lamb brush chromosomes. Now, if you talk about lamb brush chromosomes, then they are the sites of active RNA synthesis. So RNA synthesis or transcription increases tremendously, so that all the required mRNA and all the required proteins are synthesized in the egg in diplotin stage itself. Then we have oogenesis in insects. In insects, we termed as oogenesis as meiotic oogenesis. There are several types of oogenesis in insects, but the studies have focused on those insects, including Drosophila and moths, that undergoes meiotic oogenesis, in which the cytoplasmic connection remains between the cells produced by the oogonia. Now, in all the organisms, oogenesis is the basic process in which the egg has to accumulate all the things which will be required immediately after fertilization. Amphibians do it by Lamb-Rush chromosome. Insects do it by retaining the connection between the developing oocyte and the nurse cells, so that the developing oocyte can get all the material from the nerve cells. The oocyte of mereostic insects do not pass through transcriptionally active stage nor do have Lamprecht chromosome. Rather, RNA synthesis is largely confined to nerve cells. So, in case of insects, what happens? The oocyte remains connected with the nerve cells. There is no Lamprecht chromosome in the oocyte in insects, but the only thing is the nerve cells which are connected to the oocyte actively secretes RNA and then transfer this RNA to the developing oocyte. And these RNA made by those cells are actively transported into the oocyte cytoplasm. Oogenesis takes place in only 12 days so that nerve cells are metabolically very active during this time. Nerve cells are aided in their transcriptional efficiency by becoming polytic. Now, oocyte don't have Lamprecht chromosome. Oocyte don't have any specialized mechanism to increase their transcription rate, but yes, nerve cells have polytene chromosomes. Now, polytene chromosomes are also giant chromosome like Lamprecht chromosomes, and in polytene chromosomes we have, instead of 2 copies of chromosome, they replicate until they have around 512 copies, and 15 nerve cells pass their ribosomal and messenger RNA as well as proteins into their oocyte cytoplasm. So this is the slide which shows how basically uh, nerve cells and oocyte is derived in insects from a single oogonia. You can see this is a single oogonia. It divides into 2, then again it divides, then it divides into 4, ultimately it divides into 16 cells. Now the thing you should notice here is you can see this red structure which is labeled as fusome. This fusome don't divide, it moves only to single cell. Like in this case 2 cell stage, this fusome will migrate to cell number 1, and then again in 4 cell stage this fusome is retained by cell number 1. Ultimately what happens when the 16 cells are formed, the fusome is present only in cell number 1. All the other 15 cells don't have fusome, but you can see that all these cells retain connection with the cell number 1. Why? Because it has fusome. So basically that cell which has the fusome is destined to become the oocyte. Rest all other cells will become nerve cells and they are termed as cytocytes. Fine, this is oocyte and others are cytocytes. So basically what happens, the fusome is retained by only one cell and that cell is termed as oocyte. That cell will become the egg. Rest all the 15 cells are going to function as nerve cells and they will have polytene chromosomes. They will undergo active transcription and they will form large amount of RNA which will ultimately be transferred to this cell number 1, uh, which needs these RNAs for uh, functioning after the fertilization.